In this video, we will continue our discussion from our previous videos where we have seen that how how one can replicate or biomimetic pancreatic cancer on a chip. And in this figure or in this video, we will validate the results that we got from this biomimetic pancreatic cancer on a chip in the in vivo mice models. And we will use two kind of mice models. First one is the generic mice model, and another one is this genetically engineered mice model that has most of the human characteristics. And here they have also used the same reporters. In the previous videos I have by mistakenly said that they have used anti-CD33. However, they used anti-CD31 to see the epithelial cells. And so let us start with this figure with the panel A. So what they did is they subcutaneously injected the tumor on day and considered it as day 0, and after 14 days they have observed the tumor cells, epithelial cells and apoptotic factors, and they can see that the epithelial cells which are close to the tumor cells has this has the signals for this apoptotic factors, which is represented by this white color. And they can also see this in this panel A, the tumor cells trying to ablate the endothelial cells by releasing the apoptotic factors, and these are the individuals images. And now to validate whether the ablation has been done by tumors in the same manner that has been observed in the biomimetic, that has been observed on the pancreatic when they did the experiment of the pancreatic cancer on a chip using FBS as a chemotactic agent. So for this they check this for this they check the images. For this they also check the images, and they have found the evidence that uh, the pancreatic cancers trying to ablate endothelial cells in a similar manner. For example, in this that is arrow that is represent the red arrow that represents the uh, progress of this ablations of these endothelial cells, and they can see that there are some collagen 4s present is there. In this case the white color represent the collagen 4. So the collagen 4 present is there, and they can see the depletion of the endothelial cells uh, at this site. And whereas if we see at the near the yellow mark, we can see that the complete ablation of the endothelial cells has been observed, and this can also be seen in this individual figure. So uh, from this they get the results that are similar to what they have observed in the this pancreatic cancers, biomimetic pancreatic cancers on a chip. Next, they decided to check further in order to further confirm that these observations are bona fide. They decided to do this experiment on genetically engineered mouse model, which has the characteristics more similar to the human beings. In this case also they can see that the tumor cells trying to ablate the endothelial cells. From this panels we can see that so in the arrow that is marked by the red arrow that is shown the collagen presence. However, there is no, or there is a decrease of endothelial cells that can be seen here, that endothelial cells are decreasing, the collagen is also decreasing, and whereas the pancreatic tumor cells are trying to ablate those depleted area by endothelial cells. And this is the DAPI to check, to confirm that these are the bona fide cells, as DAPI stay in the nucleus. So from this they further substantiate their observations and made a point that pancreatic cancers can biomimetically replicate on a chip, and that which has been validated by two kind of mouse models experiments. First one is by this genetic mouse model experiment. Second one is by this genetically engineered mouse model experiment. So I hope you are now somewhat convinced that the pancreatic cancers can be replicated on biomimetic chip to check their hypovascular properties or migration properties. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.